Hello everybody, welcome to a module on adaptive immunity. We're going to do an overview. Adaptive immunity is both cell-mediated and antibody-mediated working together, but there's more in common with the two than there are differences between the two. So this module is about what is in common between, or what is not identical, but what is similar between cell-mediated and antibody-mediated immunity. And what we'll do is we'll go through, and of course there are a lot of differences too, or else there wouldn't be different things. So we'll go through and we'll decide which of these elements, A through J, is a function of cell-mediated or antibody-mediated. And some of these will be both. They'll be overlap. And keep in mind, and you'll see this over and over again, antibody mediated is also called by an ancient name. It's called humoral mediated antibody. So if you hear humoral, then they're talking about antibody mediated. That's how we'll test ourselves once. And then there's this idea of selection, and we'll go through these three things, really simple. And we'll decide if these are positive selection or negative selection. And this, my friends, is the second part of this video. We're going to talk a little bit about T-cell testing or T-cell maturation, because we left it a little open. We left a little hanging thread back a little earlier when we were talking about the glands and the organs. And we said B cells are produced in the bone marrow and matured in the bone marrow. When they come out, they are ready to go. T cells are produced in the bone marrow, but when they come out, they are not ready to go. They have to go to the thymus to be matured. And this is what we're talking about. We're talking about the testing they have to undergo, the rigorous testing that the T cells have to undergo in order to succeed and graduate and move on to become mature or immunocompetent T cells. And that happens in the thymus. So, how many times have we seen this slide? We're now finally, ta-da, we're now finally on adaptive immunity. And we're going to be super specific. 2 elements to adaptive immunity that you're really going to want to never forget. It's specific to one antigen and one antigen at a time, and that adaptive immunity always remembers. Thus, vaccinations and the need to hopefully, or I should say the lack of need to have anything more than 2 shots of the vaccination, ideally, theoretically, for your entire life. That's what it's all about, theoretically. The 2 elements to adaptive immunity, if we separate them out, are the humoral or the antibody-mediated, and that's all about B cells. B cells producing antibodies that will go to work battling against antigens or foreign invaders. Or cell-mediated immunity, which is cytotoxic T cells that go to work battling against cells that have become infected by pathogens, microbes, bacteria, viruses, so on. Or your own cells that have become cancerous. That's also a good target for cell-mediated, and a really important target for cell-mediated immunity via cytotoxic T cells. An antigen, we throw this word around all the time. What is an antigen? Well, an antigen, if you break it apart, excuse me, would break into 2 parts. An antibody generator. So an antigen is something that generates an antibody. They're usually some sort of macromolecule proteins or lipids, but more likely glycoproteins or glycolipids or lipoproteins. What is that? It's a combination of a lipid combined with a protein. What's a glycoprotein? A sugar combined with a protein, complex macromolecules. And they're usually affixed to the cell wall or to the cell membrane on the outside. And they're called epitopes. That's another word for it. So the epitope is the actual protein displayed, or the glycoprotein, or the lipoprotein, or the polysaccharide. They can be either on the outside of the antigen, or the microbe, or the bacteria, or the virus, or they can be kind of loose free. So here they're shown attached to the bacteria. Call this a bacteria. But the same epitope could be launched out into tissue, and the same antigen could be freely floating through tissue. And we'll talk about exogenous and endogenous antigens and how we battle both of those. What parts of bacteria are most likely to be antigenic, are most likely to generate, and when you're antigenic, you generate an immune response. So what part of a bacteria, you might ask, is most likely to be antigenic? And it's going to be the parts of a bacteria that are most different from you. So if you say to me, I think that nuclear proteins are going to be really, really the part of a bacteria that's going to be foreign to you. So, well, it's probably not going to happen because bacteria don't have nuclei. So, okay, well, maybe it's probably then endoplasmic reticulum. I said, well, endoplasmic reticulum is really common between all organisms. Bacteria have endoplasmic reticulum. I have endoplasmic reticulum. You say, oh, well, it must be mitochondria. It must be something on the mitochondria. I go, no, bacteria have mitochondria. I have mitochondria. And then you go, what about if it's on the cell wall? And then I say, yeah, now we're getting somewhere. Because bacteria have cell walls and I don't. That's a good thing to make antigenic. That's a good thing so you really cut down on the amount of confusion. Oh, does this belong to... No, it doesn't belong to me. I don't have cell walls. It belongs to the bacteria. Or you say, okay, let's make the antigen something on the flagella, the tail that moves bacterial cells. That's a really good way to do it, because the only cells in the human body, you may not even have them. The only cells in the human body that have flagella are sperm. So that's a really, really good way to differentiate. Make the antigenic component of a bacteria some part of the bacteria that's very different from some part of your body. That'll help you out a lot. Okay. Adaptive immunity is going to take place inside mostly, excuse me, inside the lymphatic system. So you're past the innate defenses that you've been run over. Your innate defenses have run over. We're no longer talking about your integument or your mucosal membranes. You've been attacked, and these things have penetrated your body into the deeper tissue. So you want to bring the fight out of the tissue for the most part to start off to some degree and into the lymphatic system. So how can these antigens, once they get past the innate defense mechanisms, once they get past the integument, past the mucosal membranes, how can they enter the lymphatic system? Well, I'm glad you asked, because there's three ways. One, if they enter directly into the blood, which is very, very likely, they'll be filtered at the spleen by the white pulp of the spleen in the sinusoidal capillaries that just allow everything. Essentially, when the capillaries get to the spleen, they just dump the contents into the spleen, let it all be sorted out, and then recollect it and bring it somewhere else. Sinusoidal capillaries are so open. Everything comes out essentially into the spleen. Spleen is one of the most highly vascularized organs in the body. If you rupture your spleen, you can bleed. You can die from internal hemorrhaging if you rupture your spleen. You'll see a line coming in here, and this is lymphatic system. Remember, this is not afferent. There are no afferent vessels bringing lymphatic lymph into the spleen. That does not happen. These are efferent. Let me draw you a one-way arrow. These coming out of the spleen are one way in the direction of the arrow that I'm drawing, leaving the spleen. What enters? Well, blood vessels, not shown here, but all these blood vessels, especially the splenic artery, which would come about right about here, off the celiac trunk. It's a major branch off the celiac trunk. So let me get rid of this now. Get rid of all that. Let me just show you that. Let me change to red. I'll make this a little bit more color friendly to you. The arteries are going to be feeding the spleen. The splenic artery and other smaller arteries are going to be feeding the spleen. So the blood is afferent. I'm trying to make these arrowheads here. The blood is afferent into the spleen, and then it's filtered in the white pulp, which surrounds the central artery. So there's B cells, T cells there, and then efferently coming out of the spleen. And I'll go back to that sort of what was it, cyan? I'll go back to that cyan. Efferently is lymph, because every organ produces lymph. Every tissue produces lymph. The blood that goes into the spleen is filtered and bulk flow. Less fluid goes back into the sinusoidal capillaries than came out, which leaves a little extra fluid. That's picked up in lymphatic capillaries to lymphatic vessels and afferently into lymphatic trunks, and then up it goes, and it's going to pass through all the nodes, which is point number 2. Once the lymph enters the lymphatic system and it travels back towards, and we know it's coming up here, and it's going to enter back into the blood system eventually, but as it's moving along, let's say it was in the left lumbar trunk down where I have the cursor now, as it's moving along the lymph system back towards the heart, back towards the blood vessels, it's going to run into these lymph nodes. It's going to be filtered. At each one of these lymph nodes, these little bean-shaped lymph nodes, we've talked about this, it's going to be filtered. Or thirdly, if you inhale something, or if you ingest something as good examples, you're going to run into malt. You're going to be running along a mucosa, the mucosa that line the GI tract, the mucosa that line the respiratory tract, the mucosa that line the urogenital tract. They are all lined with MALT, mucosa-associated lymphatic tissue, probably most well known as tonsils in the case of the palatine and the lingual, sublingual tonsils, right? Among others, and the other tonsils that exist throughout. And so those are MALT that are being kept. They're there to filter out items that are ingested or otherwise enter the body, and MALT filters them out, and then from there MALT can pull pathogens and antigens into the lymphatic system, and that's what you want. You want these things on your home turf in the lymphatic system. What is a complete antigen? It's one that creates two effects. One that is bound by antibodies, and the one that can be bound and then can also produce, or it can activate the immune system or the lymphatic system. So a complete antigen can be bound by an antibody or by some sort of receptor, and then it can evoke the huge response that you want. It can evoke quadrillions of antibodies being produced against it. It can unleash trillions of cytotoxic T cells. That's a complete antigen. A hapten, and hap means half, really, haploid. So a hapten is one that's reactive, meaning it can be bound to, but alone a hapten cannot induce that huge immunological response. A hapten, a good example is poison ivy or poison oak oil, urushiol, and it can enter in, and on its own it is a hapten. It can be reactive, things can bind to it, but it's not going to create a huge immune response unless the oil binds to something else. And if a hapten binds to something else, then you get the full immune response. So hapten, think of it as these little beige T's, miniature T's, and then some larger carrier protein. And when they bind together, this on the right is a full complete antigen. So the hapten bound to a carrier protein, that can not only be bound, but that can invoke the full immunological response. So, telling you there's a lot of antigen. There's something like 2 million bacteria, a million or 2 million different species of bacteria alone that have been found, with the idea there might be 10 or 15 or 20 million more species of bacteria we don't even know about. We may never know about. Each bacteria could have thousands upon thousands or millions of antigens. That's a lot. How are you going? And by the way, you are going to identify every single antigen, and you're going to be able to, by name and by shape, identify every antigen that's pretty much ever thrown at you your whole life. Which, if that doesn't blow your mind, I don't know what will. So let me ask you, how is that possible? How is that possible that you can identify any antigen that's possibly out there in nature? Well, let me answer a question with a question. How many people in this world specifically can you see and not just say, oh yeah, that's that guy that you remember? No. How many people can you specifically see and name correctly? First name, last name. Forget about middle initial, but first name, last name, boom and boom. I know that person. That is, boom, Jane Doe. Okay? How many? And when I first saw this question, I immediately thought 10,000, which is true, the absolute highest end of the range. I mean, that is off the charts. Somebody with a memory of faces and an incredible memory. Peak of their life. Somebody who just hobnobs and sees everybody and knows everybody. The range is actually 1,000 to 10,000 people that you can identify. And this includes anybody. Any actor, not just friends. Anybody that you can see a face. An actor, a music personality, TV personality, a friend, a family member, anybody. Anybody that you can see their face and you can identify it by first and last name. 1,000 at the low end for those that kind of, maybe just keep to themselves a little bit more or don't watch a lot of TV or social media, to 10,000 for a bigwig moving influencer, okay? With the average about 5,000. So let's just call it 10,000, okay? You can name about 10,000 people in this world, specifically by name. And really you're talking about specifically by name because an antigen is a very specific structure, and your immune function in your lymphatic system, the immune function of the lymphatic system, can recognize more than 10,000 specific antigens. How many more than 10,000? Well, a lot more than 10,000. More than 100,000. More than 1 million. This is individual molecules that you can recognize. More than 10 million. More than 100 million. Your lymphatic system and the immune function, adaptive immunity, can recognize 1 billion different molecules. Specifically and accurately, 1 billion different molecules. Can you even imagine I say to you, I'm going to sit you down and I'm going to put 1 billion faces in front of you and you're going to name every single one of those people correctly? That, my friends, is what your immune system can do. That is what your immune system can do. That is the phenomenal human being that you are, that your immune system can do that. And to give you an example, if you want to talk about how many words there are in the world, roughly, you take the amount of words in an average language like English and the amount roughly of spoken languages, you multiply these two together and you get about 1 billion. What I'm saying to you is an analog of what the immune system can do in terms of recognizing 1 billion individual different antigens correctly is the equivalent of you saying, in addition to naming 1 billion people accurately, it's also saying any spoken word on planet Earth, you know what that word is and you know what it means. Any language, any spoken word in any language, and you know exactly what it means. Can you imagine what your value would be as a human being if you could understand every word spoken by every human being on the planet? Well, that's the value of your immune system, because it can recognize every antigen in nature. And anyway, so if you can recognize 1 billion proteins, how is that possible? Because the receptor has to match. If you can recognize 1 billion proteins, how's that possible when you only have about 30,000 proteins in your body? And one gene makes a protein roughly. You only have 30,000 genes. And the vast majority of the proteins that your genes make, I mean the wild majority, are not involved in the immune system. They're involved in moving muscles or contracting muscles, all these other functions in your body that you need to do. How about neurons alone? How many huge neurons are there in your body that have nothing to do with immune function, or muscle cells that have nothing to do with immune function, or connective tissue cells that have nothing to do with immune function? So only a small fraction. How do you get just a few thousand proteins dedicated to the immune system to recognize 1 billion different things? And the answer is there's only 52 cards in a deck. But if I dealt out a hand of 5 cards, does it make sense that I could make a lot more than 52 combinations? So think of it this way. Think of the individual proteins that you make as being like a card, but think of a receptor that you build. You scramble a bunch of different parts to build a receptor, like the 7 of hearts, the queen of spades, the 2 of diamonds, the 3 of spades, and the 4 of clubs. That's a hand, but you can mix that up. And that's what a receptor is. It's a mixture of different pieces. And when you start doing that, the combinations are incredible. So how many could you make? How many 5-card hands could I deal to you? If I dealt you a 5-card hand, and we had a full deck, and I dealt you 5 cards, you looked at it, put it down, and I dealt you the exact same 5 cards, what would the odds of that be? Well, the answer is about 300 million. And so if we go to a 6-card deck, we're way over 1 billion. Way over 1 billion. Okay? That you could, different hands that you can make. So that's how you do it. Just like you shuffle the cards together in combinations, you shuffle the proteins together in combinations to make 1 billion different receptors, each one capable of finding a specific antigen and binding a specific antigen. And that's going to be important. But since you have 1 billion different receptors that are combined, does it make sense you're not going to carry massive amounts of each one? It's just not going to happen. So you carry very low amounts of each one, which is okay. But when you're invaded, you're not invaded by a couple of things. You're invaded by billions upon billions of things, i.e. one species of bacteria that exists in billions on the head of a pin, that can duplicate every few hours to become 2 billion, 4 billion, 8 billion, by the end of the day, somewhere around getting close to 500 billion or so by the end of a couple of days. Okay. And so you have to know which one's the correct fit from this store where you just have a few, and then you've got to be able to amplify. And that's what the adaptive immunity is all about. You've got to pick the right one out of the billion that you have, where you only have a few of each, and then pick the exact right one, which you're really good at, and then say, okay, this is great. We have 1,000. We're going head to head against 300 billion right now. We're going to need more than 1,000, right? We're going to need 300 billion head to head. So let's crank it up. And that's a huge element called clonal selection. And we'll go into that in a little bit more detail, because you really don't want to go head to head with a few thousand against hundreds and hundreds of billions, because it won't work out well. Okay. And that's that cloning. And what we really want to talk about is activation. And I'll cover it briefly here, and we'll review it in the next 2 modules. But you are inactive as a body cell, and inactive. So this is an example of an inactive. This is cell-mediated immunity here. Could be, I could do one for antibody. Don't worry about it. You have an inactive cytotoxic T cell. That means it's sitting around kind of waiting for its cue. It has to be presented an antigen. This inactive cytotoxic T cell works best when this infected body cell presents it with an antigen. And if you present an antigen like this infected body cell, or a macrophage, or a dendritic cell, or a B cell, there are 3 types of antigen presenting cells: macrophages, dendritic cells, or B cells. They're called APCs, antigen presenting cell. If that antigen presenting cell presents the antigen to you, and the antigen is this red ball, hands it to you, that really triggers the immune system. And that allows for matching with this receptor in this sort of orange half shell right here. That allows for binding, and that allows you to identify this receptor out of 1 billion. This receptor on this cell is the correct one to match the antigen. Now you know to make a billion or so copies of this one specific inactive cytotoxic T cell. And that's what this clonal selection is doing right here. And there's a little stimulation by these important, co-stimulation by these important chemicals called interleukins, and so on and so forth. Okay? And what's happening on the bacterial side or the viral side, what you're fighting against, is absolutely explosive, out of this world exponential growth of a bacteria or a viral particle via mitosis. So how are you going to battle that? You're going to battle it with explosive exponential growth of white blood cells via mitosis. That's the only way to battle it. You got to go head to head, which means you have to make this cytotoxic T cell that's now activated by binding and co-stimulation. Now you got to make billions of copies of it. Or if this is a B cell, you need to make billions of copies of it too. Okay? And how can I explain this to you? Let's say your immune system, we know your immune system's an expert, is an absolute dead-on expert in everything to do with antigens. Let's say you are the world's greatest expert in diamonds. You know every single thing about a diamond. You can recognize a billion different features of a diamond. Carat, clarity, color, all those things. History of diamond. You know every single thing there is to know about diamonds. I come to you because you're the biggest expert. I say, hey, I got this crazy diamond that I really want you to tell me all about. I think it's so rare, you're going to be blown away by it. I give you a little, I say it's from here, but it was found there, but it was cut this way. And you're the world's biggest expert in diamonds, and you're all excited. And I'm like, could you look at this diamond? It'd be really great. And you're really fired up. And you say, I'll do it because I'm the world's expert. And I shake your hand. I say, God, I'm so thankful. Thanks so much. I really appreciate it. And you look at me and you go, okay, so where's the diamond? And I go, oh yeah, the diamond, somewhere in Europe. Yeah, somewhere in Europe. Not really sure, somewhere in Europe. You can find it though. And you go, okay, I could find it. I could call up 500 of my best friends that owe me a favor and have them, and we wouldn't be looking, we would know which sort of cities, Amsterdam, you know, huge in the diamond mines. Some other Dutch and Belgian cities are huge in the diamond industry. You know what places you can go to, but does it make sense? That's going to take a lot of your time. You can do it. It's going to take a lot of time. What if on the other hand I said, hey, okay, I'm ready. Show me that diamond. What if on the other hand I went and I presented it to you? I literally just put it right in your hand, as this image on the right shows. Does it make sense that that's going to save? I'm not saying you can't scour Europe and find this special diamond, but I want you to think about somebody presenting it to you and how much quicker and more efficient the process is going to be. And our immune system thrives on APCs, antigen presenting cells, presenting antigens to immune cells, T cells and B cells, that are sitting in the lymph nodes or in the spleen and the secondary lymphatic organs, just waiting to see and check. So when you present these antigens to them, everything just gets going so much faster and more efficiently. Okay? And don't forget that you clone, there's really 3 types of cells. There's 2 types of T cells and B cells. Cytotoxic T cells are the effector molecule, or the effector cell, the effector cell. And cytotoxic T cells are absolutely dyed in the wool, the #1 component of cell-mediated immunity. B cells are the #1 component of antibody or humoral-mediated immunity. And helper T cells are critical to make both work. Helper T cells are just that important. Neither process will work without them. Cytotoxic specific to just cell-mediated. B cells specific to just antibody. Helper T cells do both. You need to clone all 3 of these to really survive the next hour of your life or 8 hours of your life. You need to clone all 3 of the correct ones. So you're cloning them all. Okay? Really important to keep that in mind. And this clonal selection is going to occur in the secondary lymph organs, the lymph nodes and the spleen, because that's where the APCs, for the most part. So I'm not saying that a immunological cell can't stumble upon an antigen and bind to it and do something. That can happen. But it really just works best when they're presented by an APC, when an APC brings it to the immune system, to the lymphatic system, to the lymph nodes and the spleen, where the T cells and the B cells are posted up and waiting in a reticular network, like spiders wait in a web. That's what the spleen and the lymph nodes are. It's a web, reticular connective tissue, with B and T cells sitting inside it, just waiting. And the APCs come and they present the antigens to the B and the T cells, and that kicks everything off. And don't forget, too, there are two lines going on here. You clonally select the effector line that I just talked about, the cytotoxic T cells that go out and eat everything in sight, or the B cells, which are called plasma cells, that go out and produce all the correct antibodies. But there's two lines. The second line are the memory. The effectors go out right away, they go to work, and they aren't going to last more than a few hours to a day at most before they're killed in their job, killed in action. It's these memory cells that don't go to work. They hide to the side and they wait for the second infection, the third infection. And that, my friends, is what vaccination is all about. That's immunological memories, those memory cells. Because the next time you get infected, it's not going to take you 6 or 7 days of agony to mount the right response. You're going to mount it in hours. You might not even know you're infected, or you might get a little, oh, I don't feel so good. Hour later, you feel great. Why? Because you ramped up to 1 billion active B and T cells in a matter of hours, not 4 or 5 or 6 days. And that's what that memory line does. Okay, APCs, presenting the antigen. How does it work? If the antigen is exogenous or free in the tissue, this is a case of an antigen, a bacteria free in the tissue moving around, you're going to go through this phagocytosis. And this is going to be an antigen presenting cell, most likely a macrophage, but could be a B cell or a dendritic. You're going to endocytose it, bring it in, phagocytose it. You're going to piece it into the antigen pieces. There's a rectangle, a diamond, and a square, what have you. And then these MHC, major histocompatibility complex molecules, that are so unbelievably important, are going to grab the pieces and they're going to present them. The MHC molecules are going to insert into the plasma membrane and they're going to point the antigen. So this diamond is a small part of this molecule. It's an epitope of this molecule, but it's definitely important. And when this gets presented out here on the surface, this becomes an APC, this big cell. And that APC is just going to take off. Where's it going? Right to the secondary lymph organs to show it to B cells and T cells. Say, here, check this out. And that's going to trigger everything to get going really, really quickly. Okay. And if the antigen is endogenous, meaning inside, why would that happen? Well, a viral particle that infects a cell. This cell's infected. This cell's in trouble. This cell's going to die. But before it dies, it takes one for the team, and it takes the viral particle inside of it that is killing it, and it digests it again as it's dying. It just takes it out as it's dying, so the cell is dying. But it does break it apart, and another MHC complex, MHC1, is going to bind to it. And again, same thing. The MHC1 molecules are going to insert in the membrane and present this antigen, which is the red ball, which it's an epitope from this microbe or this antigen, and it's going to present the specific antigen outside. This is also just became an APC. Where is it going? Right to the lymphatic organs, the secondary lymphatic organs, and it's going to present it to a B cell or a T cell. Okay, and that's how that works. And then this testing, which is really important. We're going to take a few moments to really understand this testing. This is inside the thymus, and this is a selection that we really have been avoiding, but now we really need to talk about it. So here's an immature T cell, okay? And this immature T cell right here, I'll put a little arrow, immature T cell. It's going to create these proteins. It's going to build these proteins on it. It's going to build a CD4 molecule, which is in red, a CD8 molecule, and a TCR. What does a TCR stand for? T cell receptor. Those are the three components that it has to build. Part of its maturation and development is building these things. So it's going to build CD4, CD8, and a TCR. Then there's going to be this testing that's going to come along. This is positive selection. And what happens? Well, this MHC2 right here is a part of this molecule. And these are epithelial cells, by the way. They're doing the testing. The epithelial cell brings along an MHC2 molecule because that's what antigen-presenting cells bring. And it brings it along and it sees if it can bind to the CD4 of the T cell. And if the CD4 of the T cell makes a nice match to that MHC2, that's good. That means you're going to get a nice coupling like a nice lock and key fit. It's not about the receptor and the antigen. That's a separate binding. But it means you're going to get these two cells locked together so that the receptor can take a really good look at the antigen. It's going to slow things down and really bind these cells together if the CD4 can bind to the MHC2. Also, the MHC1 has got to bind nicely to the CD8. And if that happens too, the cell's in good shape. The cell will move on to the next step, this 2 moving to the left. And by the way, this is Ninja Nerd's work here, as always. What happens if this T cell can't, if the CD4 can't bind nicely to the MHC2, or its CD8 can't find that MHC1? It gets targeted for termination. It will be killed. It will undergo apoptosis and kill itself. Let's say you move on now. Kind of important. The CD4 right here in red, I need you to know, the way Ninja Nerds draws it, becomes this red molecule right there, this red fork. And the black CD8 becomes that black fork. You see this nice binding? The CD4 in red makes a nice binding to the MHC2, which holds it in place, which is nice. The CD8 in black, this black fork, makes a nice binding to this MHC1 and holds it in place. And what is it letting it do? It's letting the TCR, which is now here, see an antigen. So the antigen is now this dot. The TCR is now these here. And now you're getting a really good look. And you're really checking to see if that TCR can find that antigen and match it. Except, here's the deal. A self-antigen. I'm going to get rid of all this ink and I'm going to highlight this right here. This is a self-antigen. This epithelial cell is putting you through the hardest test. It's bringing you an antigen over here that's a foreign antigen, and it wants you to bind to it with your TCR. It wants one of your TCRs to bind it. It's a foreign antigen. It wants your TCR to bind it. Say, hey, this is foreign, I want to bind it, which is great. Over here, this is great. Another form of positive selection, which is good. You'll pass the test. But it's going to throw you a curveball. It's going to heave this self-antigen. I'll just keep circling it. This self-antigen at you. And it's going to want you to go, oh, I'll bind that. But you don't want to bind it. You don't want your TCR to bind a self-antigen. That is a one-way open door ticket to autoimmune disease. You do not want to do that. You want to not bind your own antigens, and that's where you're being tested. So this is negative selection right here. They're hoping that you say, no, I will not bind this self-antigen. And if you do bind it, you're going through the apoptotic pathway again. You'll be killed. But if you can pass the negative selection, but at the same time, if you can positively select over here and bind a foreign antigen properly, that's a second positive selection, and you'll move on to become a mature T cell. Okay, so I'll put that right there as just one I went over right there. Can your CD4 recognize an MHC2? Can your CD8 recognize MHC1? But can you not take the bait? Can you avoid the self antigens? Okay? And then to finish off, what will happen, this T cell at the very end over here will have CD4 and CD8, but it'll randomly shut down one of those two. It'll shut down CD4 and just explode in CD8 all over the place. And that makes it a CD8 cell or a cytotoxic T cell. Remember there are two types of T cells? It's all about whether CD4. Here, Ninja Nerds has shown CD4 proliferating. Random. Totally random. All the CD8s go away, and on this molecule cell, all the CD4s proliferate all around it. That makes it a helper cell. If the CD4 happen randomly to proliferate, it becomes a T helper cell. If the CD8 proliferate, it becomes a cytotoxic T cell. Just something to think about. And again, anytime you fail any one of these selections, you go in the can. And remember, 98% of T cells fail. Only 2% go on. Okay? And if you do, you go right to the secondary lymph and you post up like a spider in a web and you just wait. You wait for an APC to say, hey, take a look at this, and then you leap into action. And that's what we're going to talk about in the cell and the antibody. All right, so let's just crank through these real fast. It involves B cells. We know that to be simply antibody. Involves T helper. We know that is both. The T helper is involved in both. Cytotoxic T is the heart and soul of just cell-mediated. Clonal selection into 2 lines, effector that go to work immediately, and memory, we know that's both. That's an integral part of both. BCRs, B cell receptors, is only antibody. TCRs, cell-mediated, is T cell is T cell receptor. That's for cell-mediated. Uses antibodies. That's the bread and butter of antibody-mediated immunity. Works best on exogenous antigens that are floating free through the tissue. That's antibody-mediated immunity. Works best on endogenous or antigens that are inside of an infected cell. That's where you cell-mediated. immunity comes in, and then cancerous or infected body cells is also cell mediated. So running long, super long, but these are so important. This module is so important. You're going to find the next one on cell mediated to be so simple because we covered all this. And then antibody will be pretty straightforward too. Those are the next two coming up. So thanks so much for sticking with it. I always appreciate your patience.